Assalamu alaikum. Today we are going to discuss lipid digestion and absorption. The items we are going to discuss today are digestion of lipids, absorption of lipids, and fate of absorbed lipids. First, digestion of triglycerides. Ingested triglycerides undergo two steps of digestion. The first is emulsification, and the second is enzymatic hydrolysis by lipase enzymes. First step is emulsification. What's meant by emulsification? Emulsification is the breakdown of large fat globules into smaller ones, and so increasing the surface area exposed for lipase enzymes. So again, what's meant by emulsification? Emulsification is the breakdown of large fat globules into smaller ones, and so increasing the surface area exposed for lipase enzymes. This is done in the mouth by chewing, in the stomach by peristaltic contractions, in the intestine also again by peristaltic movement and by bile salts. And this bile salt has very important role which we will discuss later, inshallah. The second step is digestion by lipase enzymes. We have several types of lipases. Lingual lipase secreted in the mouth, gastric lipase in the stomach, pancreatic lipase from pancreatic juice and secreted in the intestine, and finally intestinal lipase. The most active of these is pancreatic lipase. Pancreatic lipase needs several factors in order to act. The first one is bile salts. The bile salts, as we mentioned, convert large fat globules into smaller ones. This is done by lowering the surface tension. This leads to exposing large area of triglycerides to the action of pancreatic lipase. So again, the bile salts converts large fat globules into smaller ones by lowering the surface tension. And so this exposes larger area of triglyceride to the action of pancreatic lipase. The pancreatic lipase also needs calcium ions and cofactor called colipase in order to act. Pancreatic lipase hydrolyzes the primary ester bond of triglycerides, converting it into free fatty acid and beta monoglyceride. We will discuss this later on the diagram. So, what's the role of bile salts in pancreatic lipase activity? Bile salts are required for proper functioning of the enzyme. It helps in combination of lipase with colipase, and also help in emulsification of fats by converting large fat globules into smaller ones by lowering the surface tension, and this increases the surface area exposed for the action of pancreatic lipase. So now this is the triglyceride, this is glycerol, and these are three fatty acids. So this is triglyceride or triacyl glycerol. The pancreatic lipase can act upon this position. This is called alpha position or primary ester bond. This is also called alpha position or primary ester bond. While in the middle, this is called beta. Okay, this is called beta position. The pancreatic lipase can act only upon alpha position. This primary ester bond, alpha position, and this one, alpha position. Okay, so here it hydrolyzes this fatty acid and this fatty acid, releasing the two fatty acids. While this fatty acid in the beta position, the pancreatic lipase cannot act upon it. Pancreatic lipase needs colipase, bile salts, and calcium for its action. So again, it hydrolyzed this fatty acid in alpha position and this fatty acid also in alpha position, releasing two fatty acids and only leaving the fatty acid in the beta position. So this compound is called this glycerol. Okay, this is the glycerol and this fatty acid in the beta position. So it's called beta mono, one fatty acid at beta position, glyceride with glycerol. So one fatty acid together with glycerol and this fatty acid is at beta position. Now, the isomerase enzyme acts upon this fatty acid, changing its position from the beta to alpha. So it acts upon the fatty acid, changing its position from beta to alpha. So now the compound is called alpha monoglyceride. Alpha mono means the fatty acid is at alpha position together with glycerol, so alpha monoglyceride. Now the pancreatic lipase can act upon it and hydrolyzing this primary ester bond at alpha position, releasing one fatty acid and the remaining glycerol. 72% of beta monoglyceride is absorbed as such. 6% of alpha is absorbed as alpha monoglyceride and 22% is absorbed in the form of glycerol and free fatty acid. So 72% is absorbed in the form of beta monoglyceride, 6% is absorbed in the form of alpha monoglyceride, and lastly 22% is absorbed in the form of glycerol and fatty acid. So again, the pancreatic lipase hydrolyze only the alpha position, the primary ester bond in the alpha position, this alpha position, and this alpha position, releasing two fatty acids and leaving only the fatty acid in beta position. This leads to the compound called beta monoglyceride and two fatty acids. 72% is absorbed in the form of beta monoglyceride. Isomerase enzyme is an enzyme which changes the position. So it changes the position of fatty acids from beta to alpha, and the compound resulted is called alpha monoglyceride. This fatty acid is acted upon by pancreatic lipase, releasing one fatty acid and remaining glycerol. So again, the mode of action of pancreatic lipase can hydrolyze only the primary ester bond at alpha position, and it cannot hydrolyze the ester bond at the position 2 or beta position. So it removes at first one fatty acid at alpha position, releasing alpha beta diglyceride. So this is the first fatty acid, and then another fatty acid in the alpha position, releasing beta monoglyceride. Then the isomerase enzyme acts upon the fatty acid in beta position, converting it into alpha monoglyceride. And now the alpha monoglyceride can be acted upon by pancreatic lipase, releasing free fatty acid and glycerol. So again, this is a triglyceride. This is glycerol with free fatty acid. As we said, this is alpha position, this is alpha position also, and this is the beta position. The pancreatic lipase can act only upon alpha position. At first, it releases the free fatty acid from alpha position. So the remaining is called alpha beta diglyceride. Okay, and then acts again on the alpha position also here, releasing one fatty acid, free fatty acid. This is the fatty acid remaining at beta position, so the compound is called beta monoglyceride. So there is only one fatty acid remaining here at beta position, beta monoglyceride. The isomerase changes the position of fatty acids from beta to alpha. So it releases at first alpha monoglyceride, which can be acted upon by pancreatic lipase, releasing one free fatty acid and glycerol. 72% is absorbed in the form of beta monoglyceride. 6% is absorbed in the form of alpha monoglyceride, and lastly 22% is absorbed in the form of free fatty acid and glycerol. So again, the pancreatic lipase acts upon alpha position and not beta position. So it hydrolyzes one fatty acid at alpha position, releasing one free fatty acid. The remaining is alpha beta diglyceride. So it acts again upon the alpha position here, releasing more one free fatty acid. Okay. The remaining compound is called beta monoglyceride, which has one fatty acid at beta position. The isomerase enzyme acts on this fatty acid, converting its position from beta to alpha. So now the compound is called alpha monoglyceride. It can be acted upon by pancreatic lipase, hydrolyzing this primary ester bond at alpha position, releasing one free fatty acid and glycerol. 72% absorbed in the form of beta monoglyceride, 6% in the form of alpha monoglyceride, and lastly 22% in the form of free fatty acid and glycerol. Fats have a high satiety value. This is due to secretion of the hormone called enterogestrone hormone. This enterogestrone hormone delays the rate of emptying of the stomach. It inhibits the gastric motility and retards the charge of bolus of food from the stomach, so it leads to high satiety. So again, the fats have a high satiety value. This is due to hormone called enterogestrone hormone. This hormone delays the rate of emptying of the stomach. It inhibits the gastric motility and retards the charge of bolus of food from the stomach. So now we have finished digestion of triglycerides, first by emulsification and second digestion by lipases, and the most active is pancreatic lipase. And now moving to digestion of phospholipids. What do we mean by glycerophospholipids? Glycerophospholipids means phospholipids that contain glycerol in its structure. For example, we have two examples in our previous lecture, lecithin and cephalins. Lecithin and cephalins, while sphingomyelins contain sphingosine and doesn't contain glycerol. The phospholipid contains fatty acids, as we mentioned, alcohol, phosphoric acid, and nitrogenous base. Again, it was composed of lipid component and non-lipid component. The lipid was fatty acid and alcohol, and the non-lipid phosphoric acid and nitrogenous base. This is the glycerol, okay? We have here two fatty acids, this fatty acid and this fatty acid, okay? So we have two fatty acids and glycerol. This is the phosphoric acid, and the X here is nitrogenous base. This phospholipid can be acted upon by a pancreatic enzyme called phospholipase E2. This enzyme, phospholipase E2, acts on the fatty acid in position number 2. It removed the fatty acid in position number 2, releasing this fatty acid. And the remaining compound is called lysophospholipid. Lysophospholipid. So again, the glycerophospholipids, which are the phospholipids which contain glycerol in its structure, like lecithin and cephalins, are acted upon by an enzyme called phospholipase A2, which removes the fatty acid in position number 2 and releasing lysophospholipid. And now, digestion of cholesterol esters. What's cholesterol ester? Cholesterol ester is a cholesterol bound with a fatty acid. This cholesterol ester is acted upon by an enzyme called cholesterol esterase or cholesterol ester hydrolase, which adds a molecule of water, releasing fatty acid and cholesterol. So it hydrolyzed this cholesterol esterase or cholesterol ester hydrolyzed the cholesterol ester into cholesterol and fatty acid. So now we have finished digestion. So if we say pancreatic juice has a very important role in digestion, explain. This is because the pancreatic juice contains several lipolytic enzymes, for example, pancreatic lipase, phospholipase E2, and cholesterol esterase. The pancreatic lipase needs bile salts, colipase, and calcium for its action. The bile salts help in emulsification of lipids, exposing larger surface area for the action of pancreatic lipase. The pancreatic lipase then act on the primary ester bond in alpha position, releasing beta monoglyceride. Then isomerase changes the position of fatty acids from beta to alpha, releasing alpha monoglyceride. And lastly, the pancreatic lipase act again on alpha monoglyceride, releasing free fatty acid and glycerol. The phospholipase A2, or sometimes called the lecithinase. This is because lecithinase act on lecithin, which is a phospholipid, releasing lysolecithin and free fatty acid. Cholesterol esterase act on cholesterol ester, releasing cholesterol and free fatty acid. And now moving to our second objective, which is absorption. What are the end products of lipid digestion? At first, the end products of triglyceride were monoglyceride, beta and alpha monoglyceride, free fatty acid, and glycerol. So these are the end products of triglyceride digestion. Cholesterol ester was hydrolyzed into cholesterol and also free fatty acid. And phospholipids were hydrolyzed to lysophospholipid and free fatty acid. At first, the glycerol and short chain fatty acid. Short chain fatty acid means they have less than 10 carbons. Carbon atoms. Glycerol and short chain fatty acids are water soluble. They are water soluble, so they are carried through the portal circulation directly. Okay, so they are water soluble and carried through portal circulation directly. They don't enter the lymphatics. The remaining long chain fatty acid containing more than 10 carbon atoms, monoglycerides, cholesterol, and lysophospholipid, all of these need bile salts in order to be absorbed. The bile salts have two ends. They have unpolar end and polar end. The bile salts surround these components, long chain fatty acid, monoglycerides, cholesterol, and lysophospholipid. They surround all these components with the unpolar ends because they are lipids, okay? So it surrounds them, the bile salts surround them by unpolar ends and directing their polar ends outside in order to be water soluble. So this form what's called water soluble micelles. What do you mean by water soluble micelles? They are small molecules which are water soluble, formed of unpolar end directed inside and the polar end directed outside. So again, the bile salts surround these components, long chain fatty acid, monoglyceride, cholesterol, and lysophospholipid, and surround them by the unpolar end, and direct their polar endings, the polar endings of the bile salt directed outside in order to be water soluble. Now these micelles are soluble in water. These micelles are formed of bile salts together with long chain fatty acid, cholesterol, monoglycerides, and lysophospholipid. So these micelles are now soluble in water. They enter the microvilli of the mucosal cells of the small intestine by endocytosis. So they enter the small intestinal cell, where the fat digestion is completed by intestinal lipase. So the bile salts now deliver these lipids to the intestinal cell in order to complete its digestion by intestinal lipase, while the bile salts themselves are reabsorbed again to the liver by enterohepatic circulation in order to be secreted once more and act more and more. Okay, so the bile salts now deliver these lipids in the form of water-soluble micells into the intestinal cell in order to complete its digestion by intestinal lipase, and then the bile salts themselves are reabsorbed again to the liver by enterohepatic circulation. In the mucosal cells, the triglycerides and other lipids are resensitized once again. So the triglycerides and other lipids are resensitized once again. So now we have triglyceride once again, phospholipids once again, and cholesterol ester. Okay, they all bind with a protein in order to form chylomicrons. You remember chylomicrons? The chylomicrons was a lipoprotein. So it's formed of lipid part and protein part. The lipid part are now triglycerides, phospholipids, cholesterol, and cholesterol ester. They all bind with the protein part. The protein part is called apolipoprotein B48. Apolipoprotein B48. This is the protein part of the chylomicron, and this is the lipid part of the chylomicron, triglycerides mainly, together with some phospholipids and cholesterol. The chylomicrons, when formed, enter the lacteals and the lymphatic drainage to the thoracic duct and finally to the systemic circulation. So again, these water-soluble micelles, which is formed of bile salts, together with the end products of fat digestion, monoglycerides, cholesterol, lysophospholipid, and long-chain fatty acids, these water-soluble micelles enter the microvilli of the mucosal cells of the small intestine, where the fat digestion is completed using the intestinal lipase enzyme. The bile salts themselves are reabsorbed once again to the liver by enterohepatic circulation. After digestion, the triglycerides and other lipids are resensitized once again. These triglycerides, phospholipids, and cholesterol all bind with a protein, this protein called apolipoprotein B48. This is the protein part of chylomicron, and this is the lipid part of chylomicron. Both together form chylomicrons. The chylomicrons enter the lacteals to the lymphatic drainage and then through the thoracic duct to the systemic circulation. So again, this is the digestion of triglyceride. The triglyceride was acted upon by pancreatic lipase, releasing beta monoglyceride, which is converted by isomerase to alpha. This is alpha monoglyceride, and acted upon by pancreatic lipase, releasing glycerol and free fatty acid. The 72% is absorbed in the form of beta monoglyceride, 6% is absorbed in the form of alpha. Of course, these two monoglycerides need bile salts, so they will be absorbed in the form of water-soluble micelles with bile salts. While the glycerol and short-chain fatty acid, they are absorbed, 22% absorbed directly to the portal blood, so they don't enter the lymphatics. 22% glycerol and short-chain fatty acid will be absorbed directly to the portal blood. They don't enter the lymphatics. The monoglyceride, alpha and beta monoglycerides, they continue their digestion inside the intestinal cell by intestinal lipase. For example, here the alpha monoglyceride releases one free fatty acid and releasing one glycerol. The intestinal lipase act on alpha monoglyceride, releasing one free fatty acid and glycerol. This glycerol, once again, as we mentioned, resenses of all lipids takes place. So the glycerol at first is activated and then combined with three fatty acids, forming triglyceride once again. Also here, the beta monoglyceride means glycerol with only one fatty acid. It combines again, it can combine also with two fatty acids. Of course, after activation of fatty acid, it can recombine again with two fatty acids, forming also triglycerides. So now that triglyceride is resynthesized once again. Also, lysophospholipid can combine with three fatty acids, forming phospholipid once more. Cholesterol can combine with fatty acid and form cholesterol ester once more. All these lipids, triglyceride, phospholipids, cholesterol, and cholesterol ester, of these combine with the protein part of chylomicron, which is called apolipoprotein B48. Apolipoprotein B48. So this is the lipid part and the protein part, apolipoprotein B48. They form chylomicrons. The chylomicrons enter the lacteals through the thoracic duct. They reach the systemic circulation. And now finally, what's the fate of the absorbed lipids? 2 hours after eating a fatty meal, there is turbidity of the plasma. 2 hours after eating a fatty meal, there is turbidity of the plasma. This is due to the circulating chylomicrons. So these circulating chylomicrons, after absorption of lipids, leads to turbidity of the plasma. This turbidity of the plasma appears 2 hours after a fatty meal. So this is due to circulating chylomicrons. This turbidity is soon cleared by an enzyme called lipoprotein lipase, which clear this turbidity. This enzyme clears the turbidity, so it's called clearing factor. This enzyme is a lipase enzyme, so it acts or hydrolyzes the triglycerides present in the chylomicron into glycerol and free fatty acids. It hydrolyzes the triglycerides present in the chylomicrons to glycerol and free fatty acids. The glycerol is transported to the liver to be metabolized as carbohydrate, while the free fatty acids are transported by plasma albumin, transported by a plasma protein, which is plasma albumin, transported to the liver, adipose tissue, in order to be metabolized. So again, 2 hours after eating a fatty meal, there will be turbidity of the plasma. This turbidity of the plasma is due to circulating chylomicrons. This turbidity is soon cleared by an enzyme called lipoprotein lipase, or clearing factor. It's called clearing factor because it clears the turbidity. It hydrolyzes the triglycerides of chylomicrons to glycerol and free fatty acids. This glycerol is transported to the liver to be metabolized as carbohydrate, while free fatty acids are carried by plasma albumin to the adipose tissue, to the liver, to be metabolized. So finally, in our lecture today, we discussed digestion of lipids, absorption of lipids, and fate of absorbed lipids. Regarding the digestion of lipids, at first we discussed digestion of triglycerides. The triglycerides must be at first emulsified using the bile salts and then digested by lipase enzymes. Bile salts have very important role because number one, it causes emulsification of lipids, converting the large fat globules into smaller ones by lowering the surface tension. This exposes larger surface area for lipase enzymes. The pancreatic lipase need bile salts, colipase, and calcium for its action. It acts on the primary ester bond, which is present in alpha position only. This releases beta monoglyceride. Then isomerase enzyme acts on the fatty acid in beta position, converting it into alpha monoglyceride. The alpha monoglyceride is acted upon by pancreatic lipase, releasing glycerol and one free fatty acid. Then we discussed the digestion of phospholipid. The phospholipids are acted upon by an enzyme called phospholipase A2, releasing one fatty acid and lysophospholipid. The cholesterol esters are acted upon by cholesterol esterase or cholesterol ester hydrolase, releasing one fatty acid and free cholesterol. So pancreatic juice has very important role in the digestion of lipids since it contains three enzymes: pancreatic lipase, phospholipase A2, and cholesterol esterase. Regarding the absorption, the end products of lipid digestion were fatty acid, glycerol, monoglycerides, and also we have cholesterol and lysophospholipid. Regarding glycerol and short chain fatty acid, they are water soluble, they enter the water circulation directly, and they don't enter the lymphatics. The remaining end products, which are long chain fatty acids, monoglycerides, cholesterol, and lysophospholipid, all need bile salts, which surround them by their unpolar ends. The bile salts surround these lipids by their unpolar ends, while directing the polar ends of the bile salts directed outside in order to form water-soluble micelles. These micelles enter the intestinal cells, where the fat digestion is completed using the intestinal lipase, while the bile salts are reabsorbed once again through the enterohepatic circulation to the liver in order to be secreted more and more. Once the digestion is completed, the triglycerides and other lipids in the intestinal cell are resynthesized once again. The triglycerides, phospholipid, and cholesterol ester they combine with apolipoprotein B48, which is the protein part of chylomicron, in order to form chylomicrons. These chylomicrons are absorbed through the lymphatics and through the thoracic duct in order to reach the systemic circulation, where they cause turbidity of the plasma, which appears 2 hours after a fatty meal. This turbidity is cleared by an enzyme called lipoprotein lipase, and so it's called clearing factor because it clears the turbidity. Releasing it, they hydrolyze the triglyceride, releasing free fatty acid and glycerol. The glycerol is transported to the liver in order to be metabolized as carbohydrates, while the free fatty acids are transported by plasma albumin, which is a plasma protein, in order to be transported to adipose tissue, liver, and other tissues for metabolism. Hope you understood our lecture. Inshallah, We will discuss it online on Sunday.